Buenas tardes, vamos a continuar con las sesiones orales. Bueno, mi nombre es Mariela.
Good afternoon. We'll continue with the next presentations. My name is Mariela Diego. I am part of the Evolutionary Biology Sciences. We are going to introduce Angie Reyes, who's coming from the Andes University from Colombia, where she studies the genetic and neurological evolution of social behaviors and sexual mating in vertebrates. Today, Angie will talk us about the different changes in biology and the different behaviors or traits in the sexual selections. Remember that you can leave your questions in the Q&A section. And Angie, if you're ready, just let us know, and the floor is all yours. Perfect. The floor is all yours. Okay, perfect. Thank you very much. All right. Good morning, everyone. My name is Angie Reyes. I am a biologist. I have just concluded my master's degree in biomedical studies at the Andes University. I am pleased to be here to introduce you with this research that talks about the neuroanatomic divergence and its relationship with the sexual traits in populations of natural guppies. So the explanation, the evolutionary explanation in terms of the size of brain and vertebrates could be linked to the different cognitive demands from the different species. So we know that we have a specific relationship between the size of the intestine and the size of the brain. So here we know that there is a specific hypothesis of the expansive tissue that we have. And we know that this has evolved us to deliver better results in a social scenario that is going to respond to a specific behaviorist motivation, that we know that this is going to motivate also, that is going to shape also the brain. So guppies are a specific type of fish that are specifically used for behavioral studies and for mating studies. This type of fish are very sociable, and males are very well defined given their color, and females are there to establish the specific selection given their coloring of their counterparts, besides the size of the tails and the genopoid, which is the specific anal tube that they're going to use as the copulating organ. So as to talk about the motivations of this research, it is important to mention the previous studies that were performed by Coop and its Korshal et al. So here they work with the different sizes of brain that were relative to the body size. So they found some specific changes in cognitive skills amongst the ones having bigger or smaller brains. So here the ones with the biggest brains would have greater capacities, greater cognitive capacity. So these differences in brain size would be translated into some specific cognitive differences for specifically females, for the female species. So this also showed that there is a direct association between the brain size and also the different specific characteristics, such as the different physical characteristics in the body, specifically the spots, the different color spots that they would have in their body. However, we don't know yet if this could be translated into some other species. And thanks to this, we had this new project that is looking for us to understand how the brain works in nature and if there is a specific relationship between the sexual traits and the brain size as it was seen in the laboratory. So we conducted a research as to see what would be the neuroanatomy in nature, the neuroanatomical traits in nature, as to be how they could be compared to the sexual traits. In order to do so, we work with guppies here in Colombia. In Colombia, guppies have been diversified throughout the territory in different environments. We collected in total 18 different populations. So they have allotic, lytic, or intervened bodies of water. And at the beginning, in order to establish the methodology, we sampled at least 20 individuals, 10 male and 10 female samples. And here we work with the different data as different data to establish the different sexual traits. Later on, we worked on this species, we work on these animals as to analyze the brain size, and thanks to J software, we estimated the volume of different 5 areas in the brain, and the encephalic, hypothalamus, cerebellum, and the backbone. And we also had a specific scoring measuring card based on different literature, and also talking to some, or also working with some specific experts in the area, where we collected these samples. We also collected some samples, some additional brain samples with RNELA 11, where we work with the amphogyectin 1, and there was also a regulator for the angiogenesis for the 4 different populations. We conducted a PCR in real time for the first one, and the fluorescence, the resultant fluorescence values were used as to find the expressions for N1 that were normalized to the expression N1 for the house items. Initially, we used, as to establish the specific analysis model, we work with the weight of the brain, and also we analyze this between genders, between males and females. So here we have some different fixed effects, and we also control the longitudinal aspect as a variable. And then we had some linear aspects that were examined for the different volumes of the 5 different regions of the brain that were studied together, analyzed with the type of brain, the sex, the society they lived in. And here it was also included the weight of the brain as a variable. And it was also important to see, or this was also connected to the way that it was going to impact those sexual patterns that they would have to conduct their sexual selection. So this was specifically analyzed through some specific characteristics. So here we have some dependent variables and 3 different models that include the weight of the brain, the type of environment, the predation score, and also how long the body was, and some other aspects for the assessment, for this evaluation. And lastly, we conducted an ANOVA with AN1 as to establish if it was statistically different amongst populations and amongst the different genders, or the different male lines that we were working with. And this brain included, again, the body length as a variable. And now, in terms of the results, I'll share with you the different results and the different remarks that we could conclude. So here we could see some differences between the weight of the brain, and also the length of the body. And there were some significant differences between different populations and between genders as well. And we also found between sexes. And we also found differences in terms of predation. As it was expected, the type of environment and also the predation score in terms of the weight brain would be different for males and females. The red shapes here are showing us how it would be different for female bias and male bias, and how this would be in terms, or how this would be related to the weight of the brain. And the black bars are showing us the bimorphism animals that we had. Well, out of this graph, we can see that the direction of this bimorphism, sex bimorphism, it would change in terms of populations, where these populations, or where females have greater or bigger brains, and some other populations where we have the opposite situation and males have bigger brains. On the other hand, we can also see some differences in the relative volume on all of the different regions in the brain between populations and between sexes. And we can also see that there are some areas that could also vary in terms of the ecological environment, and also amongst, or having in mind, the different predation scores. And the telencephalo and the tegmoctical are bigger in elotic environment. For the opticum case and the backbone, we have some differences in terms of sexual bimorphism and the specific volume amongst populations. Now, as a matter of, let's say, discussion for this first part, we found specific differences in brain weight and some specific characteristics in terms of the populations we work with. So this could be caused by the different characteristics these fish live in. For example, the different areas of the brain are connected, or they are most of the times caused by the environment or the social case, and sometimes given the predation in some areas. On the second hand, our results are consistent with the general prediction that we have, that the more demanding environments that will require greater skills, greater cognitive skills, would select bigger brains and some specific dorsal, or let's say bigger long dorsal areas. Thirdly, we found that these guppies with greater spaces for these discussions, for these spaces, would have spaces for this predation that would favor bigger brains and would also boost neuroanatomy by imposing some specific pressure on the specific cognitive and sensorial processes. And this would also impact weight, and this would be present in both sexes, specifically for those that are selected for the male guppies that are more vulnerable to predation. Fourthly, we found different grades of sexual dimorphism as we saw throughout the graphs. So here it is also going to impact the brain size and the body and the weight of these brains. It would be likely associated to the different selection pressures that could be appointed to each of the sexes. So this could actually end up in some divergent differences, cognitive differences amongst them. The outcome effect, it is key in the movement and also the response, the split response. So by finding this dimorphism, this sexual dimorphism that is going to be greater specifically for men, for male, it is going to be consistent with the greater vulnerability for males as well. And also these males are going to develop bigger brains when predators are around. So this is going to impact the dimorphism in the tectum opticon in some specific intervening water bodies. On the other hand, females are collectors and they are more active than males. And this is going to impact the cognitive skills they have between the encephalum and the cerebellum. And there are some specific areas that have been proven for these areas have been proven for the cognitive skills. Now, in this graph, we show the allometric relationships for each part of the brain together or all together with the whole brain between males and females. So here we have the different regions and how they are going to be impacted specifically. And these parts are representing the regression out of this relationship for each one of the species. And here we evaluated the significant differences in the intersection between the regression of each part of the brain and the total weight out of all of the different populations. We found some specific differences out of this regression between populations, which is telling us that this area of the brain, it is growing as much as the brain grows, but it is going to be different for each one of the populations. On the other hand, we found also some meaningful differences between different populations. This is telling us that the regression of brain, it is not going to impact the weight of the brain amongst the different populations. Just to wrap up, we saw that the different areas of the brain are not growing evenly as much as the brain is growing. So these results could be specifically compared with different checking different studies where they found that some of the times at laboratories, this is going to change with the brain size. But they were raised in the different, but if they are raised in the same conditions as the regional ones, they are going to find a similar behavior. So this is telling us that this variation of the environment between populations could contribute to the neuroanatomical differences between species. And lastly, we had some linear modeling as to prove is these sexual patterns or sexual traits, especially having in mind this, the three main one would be associated to the weight of the brain and the different environmental traits that we also assess. First of all, we found that there was a significant difference between the brain of the size and the size of the brain, I'm sorry, and also the environment that they were in. And also there will be a specific representation between the size of the tail as well and the expansion of the black spots in their body. So this is telling us that these sexual traits and the brain size, it is meaningful, but they would change amongst the different populations, having in mind the different predation risks and also the different environmental conditions of the water bodies where these fish are in. So here we found that these black spots, body, the brain weight, and also the body length would change in terms or having in mind the specific predation conditions and also environmental conditions. Besides, we didn't find that there is a specific, a direct relationship for all the different sexual traits. And last but not least, in this slide, we see the different results of the AN1 for male and female in all populations, and A, and in B, we see AN1 for the body, for the brain weight. So when we measure this relative weight for A1, it was possible for us to predict that this could be related for all of the different populations based on the different studies with these guppy fish at the laboratory. But in our case, we didn't find any kind of evidence with this association. It is possible that this data, it is not producing, let's say, giving us more information, given the specific limitations that we have. And again, our data are limited as to establish some final results. And just to conclude, I would say that thanks to this work, we managed to assess different hypotheses in terms of the evolution of the brain. We are just complementing different laboratory studies in guppies where it is possible, where it has been possible to see the ecological impact on this fish and how this is going to impact the brain development of different species. However, this enabled us to understand how the neuroanatomical traits, cognition, and some other physiological traits might change depending on the specific environments that these species might be living in. So what's next? Or what are the next questions for us? What's next for this research line? So first of all, we need to conduct more studies using this predation scores or this predation data as to better understand how predation could actually impact brain modification or brain shaping, and also how the cognitive skills are going to be impacted given these differences. We also suggest working or researching on these selection patterns in nature and how they are going to change in comparison to the studies that have been conducted in laboratories. Finally, I'd like to thank our sponsors and also my professor, Natalia Vlog, who's been working in our laboratory at the Andes University, as well as some other collaborators that have helped us out to identify and to work with all of this field work. They are great examples of passion as to do science in Latin America, and that's why we're here today in this symposium. So thank you very much for your attention, and if you have any questions, please let me know. Thank you very much, Angie. That was a very good presentation. And let's see if there are any questions in terms of your presentation. So let's go with a question by Adriana Cero. How did you classify these predation scores, and what's the function of AN1 for the brain size? So the classification of this predation happened thanks to the literature we review and let's say the work that we conducted with experts. These experts have been working there for a long time in the Colombian territory, so we have observation. We pretty much interviewed people in the region, so they told us what kind of predators they could have seen in the region, and also some experts. So in that way, we established, let's say, a classificatory chart where we would measure predation from 0 to 5. And with the AN1, the function of AN1, in terms of the previous literature that we worked with, I think that I had this in my presentation. This has been linked to the development, to brain development. Thank you very much, Angie. We're out of time and there is an extra question. If you wanna, it's okay if you answered this question in the chat box or in our Q&A. Unfortunately, we're out of time. And again, thank you very much. Thank you.